My name is Patrícia Pelufo Silveira. I'm an associate professor at the Department of Psychiatry at McGill University, and I'm a scientific director of the Genomics and Epigenetics pillar of the Ludmer Center for Neuroinformatics and Mental Health. We know that we inherit the genetic background from our parents, so from our DNA we receive the message that is the template upon which all our functions happen. The template defines how proteins are expressed in different tissues, and they define our characteristics, our physical features, our behaviors. Functional genomics is focused on how these proteins interact with each other and how genes express these proteins according to responses to the environment or to events that happen during the life course, and how these responses influence health and disease during development. Epigenetics is defined by changes, molecular or chemical, that happen around the DNA, and this is also functional because these modifications come in response to changes in the environment or to events or stress that can happen during development. Part of the mission of the Ludmer Center is really to integrate different types of data and use these resources that are available to different researchers to make sure that we cover either the life course or different modalities to help us understand the mechanisms and the associations with mental health. The work that we do fits perfectly with the Ludmer Center because of the focus on neuroscience and the interaction between the environment and the genes, and the use of large data sets. We have collaborations in North America, Europe, and Asia. So when you do that, you really have an external validation of your findings, because you move from local and very central to your lab findings to try to find associations that replicate your findings in other contexts. Genes vary a bit according to populations in the world, so it's important that when you find an association, you replicate it in other samples. Some data is really a collaboration where scientists come and share their data. Sometimes you purchase the data too. One of these data sets is the UK Biobank that involves more than 500,000 participants from the UK with very detailed data from phenotype, their diseases, neuroimaging, genetics. It really allows the exploration of different questions using the same initial effort of collecting all this data from all these participants. The largest accomplishments that my lab had was to create a new type of polygenic score. Polygenic scores are really genetic markers of risk for disease. We move away from the traditional polygenic score that defines risk for disease to a network-based score that defines function and variation in function. We have used these scores in different contexts, different types of scores, collaborate with other scientists spread throughout the world to use this technique that was developed in the context of the Ludmer Center. The Douglas has a long tradition of studying how the environment influences gene expression and health and disease. It's also very translational. We can do more basic science work at the Douglas, but we also have important studies in the clinical setting, so we're very informed by the patients and the clinicians and the work that happens in the clinical context. We have collaboration with Dr. Celia Greenwood, in which we use polygenic scores for depression and trying to understand the relationship with the environment. Now, Dr. Greenwood is the director of the statistics genetics part of the Ludmer Center. The collaboration of our expertise is important also to try to disentangle these questions and understand better depression in this case, but mental health more broadly. I interact a lot with scientists that are more focused on basic molecular scientists and models. Translational science usually takes so long because you have a basic science discovery that is published and someone looks at it and tries to apply to the clinic. But the environment that we have here favors this interaction and accelerate discoveries. At the Douglas, for example, Dr. Cecilia Flores, Dr. Nicholas Sermakian are really trying to understand the molecular mechanisms linked to psychopathology. We are using now their platforms and their models of disease to inform the calculation of scores that can be applied to humans. It's really a translational process that we are able to develop because of this rich environment where we're all together in the same institution. With Harvard University, we are developing a study to identify biomarkers of stress in early childhood to define who's really vulnerable to stress. We know that people vary in their responses to stress. Can we identify this very early in life? Can we make a difference for development and long-term health? The polygenic score is based on gene expression data. So far, we are using what is called bulk expression data. With the single-cell genomics effort, we can go with a higher resolution and create cell-specific expression-based polygenic scores, which is amazing in terms of exploring more detail the mechanisms associated with the conditions being investigated. Open Science is an important initiative that has been coming stronger and stronger over the years. And the fact that we have today access to these different types of data set is what allows this type of work. It's impossible to collect everything and do all these studies myself. Despite the fact that the idea is already there for quite some time, there's a lot that can be done to improve accessibility and making sure that the scientists have the support that they need to share their data, because it's not simply share. The team needs to support the staff that is involved in collection and in curation of the data. If I was going to do a study with 500,000 participants, the cost would be gigantic, and the cost to purchase the data is very small compared to everything that I would need to put in place to collect the data myself. There were many discoveries in basic science and molecular neurobiology, identifying, for example, molecules that potentially could be important for mental health or mental diseases in different models, different organisms. Basic science developed quite a lot in the last 50, 70 years. Now, the application of all this knowledge to the human condition is still elusive. So this translation from basic science to clinical medicine and potentially the development of biomarkers that can identify risk, or even molecules that can inform the development of new drugs and new treatments. This connection between basic science and humans that we do here in the lab, I think it is the contribution that the Ludmer Center is doing.